Okay. Welcome to the School of Biological Sciences seminar series. It's my pleasure to have Saad Bhamla speak today about extreme collectives. Saad is an assistant professor in the School of Chemical and Biomolecular Engineering, and he's interested in becoming an adjunct in the School of Biological Sciences, right, Saad?
That's correct.
Yeah. So he's one of my favorite scientists at Georgia Tech. His lab studies curiosity-driven research involving organismal biophysics, soft matter, and frugal science. That's science on the cheap. Previously, Saad was a dean's postdoctoral fellow in bioengineering at Stanford, working with Manu Prakash. He did his Ph.D. at Stanford as well in chemical engineering with Jerry Fuller. He's a prolific inventor. He's invented several items that cost less than $1 each. A 20-cent paper centrifuge that centrifuges blood, a 23-cent electroporator that electroporates cells, and a 96-cent hearing aid. Hey, Saad, this hearing aid's getting kind of expensive. I'm just kidding. Okay, he's won the NSF Career Award, the Innovation in Med Tech Award, the Index Award, which is a design to improve life, and recently he's been a National Geographic Explorer. Let's welcome, I'll clap and welcome Saad. Hey, thanks, Saad. Let's do this.
Now, okay, thanks, David. And just confirming, I haven't accidentally muted myself. You can still hear me okay?
Yep, they're good.
Okay, awesome. Thank you so much. And thanks also to Jasmine for, obviously helping organize in the back, just making sure everything is good. I've been at Georgia Tech, as David mentioned, for 3 years plus now, and what I'll do today is share two stories from our work on collectives in two different length scales, one at single cell level, which is the image on the left, and one slightly higher length scale, larger systems, which are these worms that form blobs. So in general, our lab is focused on active matter and biological behavior in nature, ranging from a variety of different organismal systems that we focus on, from single cells to spiders to worm blobs. And I'll kind of give you a flavor of kind of our approach to understanding these behaviors and systems today in two stories. What I won't be talking about today, which is some of our engineering work, as David mentioned, is thinking about how do we enable broader access to scientific hardware and biomedical instrumentation for this for a larger community that lives. Most of our devices cost less than $1, and most recently our invention was a $1 hearing aid. The paper came out just a couple of weeks ago. Cool. With that, the work today that I'm going to be sharing is on the worm blob stuff is led by Yasmin, who's a joint postdoc with Dan Goldman and myself. And some of the single cell stuff is with Arnold, who actually was a postdoc at Stanford, but now is starting his own lab as a faculty member in physics. So the broad motivation for this work, at least the first part of the story, came about when I started my postdoc a few years ago, and I worked with Sheila Patek, who's an evolutionary biologist at Duke. And one of the questions, this was a group that I joined during my postdoc, which is a Muri-funded work. And really the question we were asking is, what are fast movements in nature, and how do organisms achieve fast movements? So when I pose this question, and if I ask you to think about what is a fast animal movement in nature, typically, the common answers that come is, cheetahs and falcons. However, now there is an emerging movement led by some of our team that we, kind of put a framework to think about ultra-fast movements in terms of thinking about accelerations. So here I'm showing you a plot with acceleration on the y-axis and length scale on the x-axis. And what is remarkable is some of these, small organisms down in the micron can achieve accelerations exceeding millions of g-forces. Here's an example. Can you confirm you can see my mouse if I move it?
We can see it.
Can somebody confirm? Okay, so here's an example. This is nematocysts in jellyfish. These are fungal spore single cells like Vorticella, and multicellular systems like trapjaw ants and mantis shrimp. These are some of the systems that Sheila studies. In when some of the work that we present in this paper a couple of years ago in 2018 is to kind of provide a framework for how universally these organisms from single cells to multicellular systems use springs and latches to achieve what in the community is also referred to as power amplification, which is to bypass the force velocity limits of their conventional muscles, such as actomyosin systems. When I looked at this, and the reason I got attracted to this is two key reasons. One is as we were tabulating this, data from over 180 systems, at this length scale, when we couldn't find as many systems, and really this gap to me revealed our gap in understanding really what's happening at these scales. And as my training is in soft matter, really to see this at the small scale is exciting that we don't know much and there's so much we could learn. And I'll share you about one story in our discovery over here. And the second, as an engineer and physicist, there's another aspect over here that's exciting is, we know that the speed of light kind of sets the limit for velocity, but really it's unclear what is the upper limit on acceleration. How fast could these systems go? Because if I hadn't known that these tiny cells could achieve millions of g forces, I wouldn't have, a priori predicted that there would be systems that could achieve such, extraordinary accelerations. As an engineer, also, it's remarkable to note that most of our fast-moving robots, by the way, cheetahs would be on the right over here in about 10G, but the fastest robots, these are flea-inspired or termite or frog-inspired robots, are basically about a millimeter. And we still, although we can fabricate at the nanoscale, all of us carry iPhones, so we know we can fabricate really at the nanoscale. Thinking about power and actuation and mechanics less than 1 millimeter for autonomous, fast-moving robots still is a challenge. So there is really a secondary motivation beyond just curiosity-driven to understand these, is to think about smaller structures and how do we actuate them. So I love cells and at this single cell, and what is fascinating to me is what they kind of lack in size, they make up in diversity and just sheer numbers. Here's kind of a whole plethora of these single cells, and you can appreciate that they come in all kinds of flavors. But specifically focusing on fast movements, kind of when I was doing my synopsis of what are the fast movements, this kind of divided into single shot and repeatable systems that I was able to characterize. So here's a couple of extreme bioweaponry. These are trichocysts from and other dinoflagellates, and nematocysts are here that shoot weapons. But really what I was looking for is something more repeatable. So Vorticella is a classic example of these that can contract. But the protagonist of today's story is Spirostoma Mambigum, kind of the organism that I decided to focus my energies, and I'll kind of explain why I was so attracted, why I was so obsessed with this organism, and continue to be for the last few years. And we study the system in our lab now. So let me introduce you to the hero of today's story. One of them is Spirostoma Mambigum. And this is really, really a humongous, a giant cell. Think of it as a dinosaur of the microbial world. It's about 4 millimeters. So look at it, swimming around, just coasting around in this petri dish of water. And typically the other single cells that people would study, some algae over here. And if I don't know the quality that's coming across in the streaming, but if you squint, you might see tiny dots on your screen. Those are bacteria. So really for a single cell with no muscles or neurons, this is a giant cell, about 4 millimeters in length. What that means is that in real life, if I hold a vial up, you can actually see these single cells with your naked eye. Really, extraordinary cells. But what is amazing about them beyond just their size is what they can do. So I hope you can focus where my mouse is, and I'm going to play this video, and don't blink, and you'll see its contraction. I'll play this video once again. And this kind of blew me away the first time I saw it. this is another natural behavior. This is now Spirostoma hanging around with its friends. And these are tiny dots that are moving in the background, or the bacteria in the cultures that they feed on. But really, the first time I laid my eyes on these cells as I was watching them through a microscope, I was just, just blown away, and had a whole host of questions about how they would be contracting, why they're contracting. A lot of these things were not known at that time when we started our research about 4 years ago. And you can clearly see they're doing it naturally. I'm not perturbing them, and they seem to be happy. They seem to be surviving these really large contractions. I'll give you some numbers for these things. So the challenge with studying something really, really fast like this is that, oftentimes if anybody's worked with single cells that can swim with cilia, is that they swim out of your field of view. And so what I did was built a setup on a microscope with a high-speed camera and a electrical simulator such that I could trigger these cells. And I really mapped out what is the intensity and the duration so that I could controllably contract these cells. So really, for the next set of data that I'm going to show you is in a regime where we are contracting the cells, they're happy, they continue swimming, but we can, contract them over and over again without killing them. But this just helps us map out really which is the sweet spot to live in, and it gives us a controllable setup. So here's kind of a first video from this, and this is high-speed video at 3000 FPS that I've slowed down. And I'll play this so you can observe. And there are a couple of interesting observations here. I believe this video is on loop. You'll notice that this one end starts contracting first, and then the second follows. There's a small delay. And what we speculate is happening is that there's a calcium wave that's traveling to synchronize across its millimeters length. You'll also observe that it's twisting from a mechanics point of view. It should kind of hint towards it relieving its stress such that the internal pressure kind of doesn't build up and it doesn't burst on itself. So inside it is a inviscid fluid. It's filled with cytoplasmic fluid, so it doesn't bulge out, but really, there are mechanics and the boundary cell membrane all interplaying. So there's a whole other lecture over here on really what is the machinery that's powering such a rapid contraction. And there's a lot we don't know. We're now at the stage we've identified the key proteins, but I'll share with you in about a few seconds what we know. The powering engine for Spirostomum is something what we call myoneums, and these form this fishnet structure. This is a confocal imaging with an antibody stain. And these myoneums form a two-component protein supramolecular spring effectively that contracts. What is really unusual about this is that it responds to calcium. And so it's different from the muscles that are powering other eukaryotic cells, such as muscles in our body, which respond to ATP, and our ATP is kind of the fuel. Here, the fuel is calcium, so it does not require any nucleotide hydrolysis for powering its contraction. It's a purely mechanochemical engine, and it's remarkable that this spring basically contracts so rapidly, and I'll give you some numbers of how much power it generates. And we're trying to figure out kind of how would such a spring actually work, and how would we actually build it in vitro for synthetic applications. So here's kind of first attempt at kinematics of this cell. What I'm showing you over here is the length of the cell, and I take the derivative first and twice to get the velocity and acceleration. And in the length, you can see that Spirostomum contracts about 60% of its length as it shrinks in about less than 5 milliseconds. To put this number into some context, you could have 50 contractions in the blink of an eye if it could do this repeatedly, but I hope you appreciate that once it contracts, it has a slow relaxation time over a second that it has to reset. The peak velocity is on the order of 0.2 meters per second, and the peak accelerations are about 100 meters per second squared. This is some of the older data. We have newer data. So that's about a order of magnitude similar to cheetahs and slightly faster. But beyond these kinematics, what's really unique about this, and for those of you who may have recognized or worked with microorganisms, a key number that's useful in this community, in this thinking about cells that are in this fluid, which is water, is Reynolds number. Effectively, the Reynolds number tells you how much inertia matters versus how much the viscosity of the fluid around it matters. And typically, we think of microorganisms to live in a low Reynolds number, which means that for these cells, when you shrink yourself down to this small level, really effectively for them swimming is at the human scale swimming in molasses. And so they have to come up with all these unconventional tricks, such as moving with flagella and cilia. And so when the spirostomum is swimming, it follows in that kind of, low Reynolds number world, which its Reynolds number is 0.01. However, when it contracts, its Reynolds number, the transient Reynolds number, jumps to 100. So you see this 4 orders of magnitude jump in the Reynolds number. And for a brief second, it jumps out of this low Reynolds number that typically confines most microorganisms and single cells. And that's interesting. And once you know that, you can kind of peek at the physical signature of this high Reynolds number or finite Reynolds number effect. So here I'm going to show you an experiment. We've got Spirostomum on a microscope between 2 slides in water. But I've done an additional thing. What I've done is put tiny micrometer bead particles, those are the dots you can see, and these are tracers. So they will follow the fluid lines surrounding the contraction to reveal what's happening in the fluid as Spirostomum contracts. So I'm going to play this video. And hopefully what you should see is these vortices that are created due to the anisotropic contraction of this body. And as the cell body contracts, it's pulling fluid around it and creating these large vortices. And this is a clear signature of a finite Reynolds number effect. I'll play that video just once again. It's just, to me, one of just so beautiful. So this starts to reveal some aspects of how it's dissipating its energy during contraction by creating these fluidic waves around it. But it also hints to something that I'm going to go into the next part of this talk, as to why spirostomum and collectives actually contract. Before we do that, we can use these fluidic estimations to kind of get a very a lower estimate of the power that these myonemes, I mentioned, this engine that's contracting, is generating by looking at the energy that's dissipated in the fluid. And if you put the numbers together, you kind of get a low estimate of this power. And typically you report this in power density because it's important to kind of normalize it by the volume of the engine effectively. And it's about 1 watts per centimeter cube. And if on this plot, I'm showing you a couple of different engines, both man-made, which is the combustion engine, and natural, such as actin assembly or bacterioflagellum or muscle, in fact. And what is remarkable is myonemes are one of the highest power density engines, comparable to kind of a 2-stroke diesel engine. Revealing again, in the first slide, I had motivated thinking about synthetic systems where we could kind of think about how to actuate small structures. This reveals to us an unusual engine that if we were able to synthetically make powered by calcium, this would be very useful, for example, in synthetic cells. If you wanted synthetic cells and artificial machines to kind of replicate the natural living parts, this is a good engine to exploit. And these are some of the activities we're exploring in our lab today. As a training in fluid dynamics, the natural thing to do for somebody with my background is, if you saw those fluidic vortices, this is a PIV image, which is just particle image velocimetry. You create these, you basically create a trace of the flow, and this is what the experimental data looks like. And you have to do some simplified linearized Navier-Stokes equation, and you numerically solve this Navier-Stokes equation with a point force function, because this is an active moving force. And if you go through the math, here's the theoretical solution, and you can see that the experiments and theory match up very well. Really what this means is that we have this biological single cell that contracts and generates these fluidic vortices, and we now understand in a numerical or a theoretical perspective how to simulate one cell and kind of faithfully capture the fluid dynamics around it at the single cell level. What we can do with this is ask how these fluidic vortices could be used to exchange information amongst these. And the motivation for this is this puzzling observation. So I'm going to play this video. And if you see my mouse, focus over here for a really surprising event that when we first saw, we just couldn't understand it, but it kind of led to this interesting discovery. So watch this video carefully. So right here, focus right here. So in case you missed it, I'm going to play this again, and I hope that it doesn't lag. You'll see that there's an instant when all of them seem to just jump apart. And I'm going to play this frame by frame. You'll see they're all extended, but between at this 30 frames per second, it looks like from one frame to another, all of them simultaneously contract. And so this was a puzzle because we were thinking about synchronization and contraction and calcium within a single cell. But how the heck did all these cells get the message so quickly to contract within less than 5 milliseconds? This is really a bizarre observation that it's almost as if information propagated throughout the system and all the cells knew, oh, we gotta, contract immediately. And so we started to think about the fluid mechanics and kind of building on the fluidic vortices that I showed you. And I'll get to it, but this is a video just to show that these cells, there's nothing artificial about confining them. Here I've just pipetted about 100 or 200 of these cells in a Petri dish. The tiny dots in the back are just bacteria. And you'll see that they naturally aggregate and they naturally form these collectives, if you will. So in this right-hand side corner, you should see them aggregate and form kind of what looks like a bird's nest. But if you watch closely, you'll see that they do these collective group contractions quite periodically, and they trigger each other. So right there, you saw that happen a couple of times. If you watch that collective group contraction under a high-speed camera, what we realize is that it's not actually a single contraction simultaneously, but it's actually a triggering event. And all of the cells are not contracting simultaneously, but it's a cell that starts to contract. That's the signal generator, but it sets off this wave and a hydrodynamic wave that the other cells act as sensors. They sense it, and there's a delay. So only the nearest neighbors feel it because the fluidic energy dissipates very rapidly, and this kind of wave travels through a colony. So really here is where we kind of are going to put together the fluid dynamics of a single contraction, where one can be a triggerer and information is propagated through the fluidic environment through these high Reynolds stresses, and the other cells are sensors and they can sense, and we've done experiments where we've kind of understood what is the critical shear stress at which they contract, and we can put together a collective model of how these cells would contract. So we can use our simulation and then ask a question about how does these waves of information propagate depending on their orientation. And here's an example which reveals that if they were aligned head to head, the information passes faster, as you would expect because of their cigar shape or antenna shaped bodies, these elongated bodies, versus if they were aligned parallel to each other. And we can effectively map out what the interaction is between these two spirostomum cells when one is a signal generator, the other one is an antenna, and characterize in space what is the most effective orientation for this information to propagate. And all the colored lines are our theoretical predictions, and these stars are our experimental predictions, revealing there's a good agreement between experiment and theory in this 2D phase space. What we can do with this is now to do these simulations at high concentrations, like I showed you, where they naturally aggregate, to really understand why they're aggregating and what are their critical densities. So in these three videos at the top, the left-hand side most one is a low density. And you'll notice that the first organism in the simulation contracts, and this wave effectively doesn't reach till the end, but it dies out. And that's because the density is so low, the information, there's gaps between them, and that message basically gets disrupted and doesn't reach the end of the colony. Here, this is about a critical density of 2 organisms per millimeter square, and you'll notice that the signal again starts from the center, but successfully manages to percolate in a fractal-like manner towards the end of the signal. And if you make these really, really dense, crowd them similar to how on a Black Friday everybody's crowding to get into a Walmart, really you're confining them onto each other, where even one cell's contraction is restricted by its neighbors that might be squeezing onto each other, or the friction is just so much, you've really made it dense. You'll notice that the information kind of dissipates throughout the center, and it's really a dense suspension. The other way to think about this is that if the goal was to percolate this information signal from the center towards the edge of the colony, it takes this meandering path, and the velocity of this information is very, very low. This is quantified kind of in this plot. And really the big takeaway, the big aha moment here, just to summarize what I've told you, is in these single cells, unlike biofilms where communication can be through biochemical pathways, here the entire communication signal propagation is through hydrodynamics, and it is critically dependent on their densities. And there is this criticality where the blue curve is showing that at low densities, the information kind of dies out. There's a critical density where the propagation is very optimal. But as you go to higher and higher densities, the signal velocity kind of falls off, revealing a really previously unknown communication pathway in these single cells for a communication between cells that are not connected to each other, revealing kind of a understanding of how cells might communicate and why these ultrafast contractions might be useful. We think of this as kind of communication at a distance in single cells that have the special ability to contract and create hydrodynamic waves. So that's kind of the first story that I wanted to share with you of collectives and behavior at the single cell. I'm going to transition to our worm blobs. And David, questions and answers in the end, is that the way?
Yeah, I think we should do it then, and then everyone can really get momentum. Let's keep on going.
Okay. So the second story is about worm blobs, and the motivation for this that we've been thinking about as we study this system is kind of thinking about how in science fiction we have this vision of how we might be able to create these active particles or ensemble of active particles that could come together and form a cohesive structure that can adapt and do some kind of useful tasks, such as locomotion, for example. And it's also, this is a spoiler, but I think Spider-Man is old enough that I don't have to tell you what the weakness for Sandman is water. So we also like to think about these systems that are ideally multi-environment and can work in air and liquids. And there's a growing community thinking about these living materials composed of these active particles in nature and kind of their cohesive properties. And all of you are familiar with David's and his team's work on fire ant aggregates and larvae. Classically, physicists and biologists have been fascinated by swarming. And I'll highlight that there's a small distinction between these systems. Here, swarming is between these unconnected individuals. They're not connected, but they show these collective behaviors. But the kind of systems that we are interested in, where you kind of find a cohesive or solid-like behavior, for example, in the fire ants, is really entanglement. In the ants' case, it could be that they're holding their legs together. And other examples within our own bodies and at single cell levels, for example, here's a slime mold that forms a slug. And really, this is a collection of single cells that have come together, coordinated by calcium weights, that kind of can achieve a collective mobility, reminiscing something of the Sandman behavior, that it has a tangible structure and is dynamic and can do something with it. And our own bodies and tissues are classic examples of these, where single cells form larger scale structures through entanglement mediated by protein networks. The reason I'm sharing all this is, if you look at the engineering side, typically the inspiration is from swarming in tissues, but the individual swarming robots are still rigid objects that communicate perhaps through Bluetooth or Wi-Fi, need to be programmed. And here's a couple of examples from Radhika Nagpal's group at Harvard, inspired by termites and ants. And kind of we are in this emerging group of folks thinking about how do we enhance kind of this interaction between these swarming robots, think about the biological side and think about the robotic side to kind of bring in some flexibility into these materials to enhance entanglement. And I'm going to go through this slide very briefly, but the motivation here is that we know enough about soft materials, and people are now turning towards soft materials rather than rigid objects to enhance kind of this entanglement that I'm referring to. So with this motivation, really we know enough about materials. We are all aware about polymer physics. Our thesis is that we need more rules on how collectives behave rather than just the material itself. And there's a emergent behavior that we need to kind of get a grasp on that we're lacking. And so we're thinking about new systems to look at. And this is for those of you who have been to Stanford about a couple of years ago when I was doing my postdoc. Actually, this is before that. So I did my PhD at Stanford. This is the Lake Lagunita. It's a lake. And for those who are familiar with California, know that there's a drought. So about 5 years I was at Stanford. It never rained there. And this is the Hoover Tower. It's a famous tower on campus. And I never saw water in this lake, but fortunate for me, I stayed over for my postdoc. And in 2016, it rained, and there's a beautiful rainbow. And everybody, over here in Atlanta, now that I'm here, when it rains, people don't even, we don't even think twice about it. But for a place that gets a lot of drought, when it rains, people just go crazy over there. And all these Stanford kids brought around boats. I don't know where they brought these boats from, and kayaks, and started to paddle in this lake. And I went to walk with the question that if you had a lake that has been dry for 5 years, I was wondering and just curious what kind of organisms would be there overnight. And what I was amazed to find are these tiny, what almost looked like, I was thinking it was dung or something. I didn't know what it was. And I took a closer look, and lo and behold, it was alive. So I poked at it with a stick, and you'll see that it's actually an aggregate of a worm blob. And so when I came to Stanford and started my faculty position here, this was kind of stuck in my mind. And for those of you who are aspiring to start your own labs, when you start your own lab, you have to think about projects. And I was like, oh yeah, worm blobs, we got to study this. So we started this project. Yasmin really championed this. We learned that these blobs are made of California blackworms. So that's Lumbricus variegatus. These are, about a few centimeters long. And here's a whole collective of it. Here's 50,000 worms, just worms, nothing. Yasmin is playing with them. And this is just fascinating, right? It's just like a bowl of spaghetti noodles or silly putty, but it's alive. There's nothing holding these worms together, but basically their own bodies, and I'll show you how they entangle. And this flows like a liquid, and I'll show you some other videos about its properties. And so we were fascinated. And you can make these small blobs, coarse them into different shapes, like Georgia Tech. So here's each individual active particle. Here's a worm, and it can swim and do a inchworm gait. It can, in a petri dish, it can crawl around. It can change its motility. So here's the head, but it can switch. So the head will go, yep, it flips. It can change directions. So it's very capable of some motility. And if you put a blob and watch it in petri dish from a solid-like cohesive thing, it can dissipate and kind of melt away like a chocolate. But if you give it some time and reduce the temperature, it can resolidify in a petri dish again, and it can aggregate into forming these cohesive solid-like structures. We think here the worm blobs are communicating with us and trying to tell us something, or maybe we're just staring at worm blobs too much. They're fascinating. They can flow through tubes at long time scales, as you would expect from some of these materials. And at short time scales, you saw that the worm blob did not splat. The worms in this are still alive, and they can withstand all these physical assaults. They're really robust when they form this collective. But here's the really remarkable feature. You put them on a dish and put a camera up on top and just watch how I'll just let you watch this, and just fascinating how they entangle with their bodies. So we were wondering how they actually knot amongst themselves and why they would do it. This is, again, a problem that hasn't been studied, and we're kind of the first to investigate this collective behavior in these systems. And it's for folks who have some recognition of polymer physics, it kind of is reminiscent of kind of reptation behavior in polymer physics. Obviously not at the same length scale. These are macroscopic systems, but really appealing to think of them as living materials. So the first hypothesis was, why form a blob? And naturally, if you recognize their living environment is in these freshwater ponds, and you saw the first time we saw them, they form this aggregate when they're out of water. Really, the natural hypothesis to make is that evaporation may be a role that's exerting a pressure and enhancing them to form these aggregates. And other organisms do it for thermoregulation. Penguins are a classic example of this, bees and other collectives also. But here's what's interesting. So I'm going to play this video just to provide some context. Here's a small worm blob, and I'm going to place it outside water, right? So this is in air, surrounded by air on a plastic surface. And I'm just watching it. So watch what it does. So what we observed is that the worm blob is kind of actively making some decisions, and I'll tell you what it's trying to do. I'll start again. You'll see it sends out kind of this arm, potentially looking for other favorable environments, perhaps with water. And if I had a small puddle of water, you would have seen the whole blob trying to migrate towards there. But it recognizes there is no water and it retreats back, and you can start to see it's a very dynamic structure. It's trying to form this hemispherical shape, and it's hinting towards what we are hypothesizing, right? That it's trying to reduce evaporation. And so you asked me, does this experiments systematically over different blob sizes, and we can quantify them using MATLAB, just the projected area. And really what we find is if you have a single worm that's n is equal to 1, the worm shrivels up and dies. But as you increase the worms, you start to see this exploration behavior. So the purple curve over here is about 1,000 worms. You see that classic increase in area that was it sending its arm out to search, but then universally they kind of go into this retrievement, and this is the area where they're forming this hemispherical cap. There are 2 points here. Number one, that if you are a single worm, the amount of time that worm survives, it's on the order of a few minutes. However, if that same worm was in a 1,000 worm blob, then its survival increases by an order of magnitude, and it can live for hours. So it's really being in a larger collective is beneficial for survival. And we can take this idea, and if you imagine each worm to be a cylinder, you can estimate its volume and how many can you pack in the cylinder. You see that how much you can pack in a hemisphere. You'll see that the experiments and theory line up. And really, the hemisphere is a surface area minimizing structure. So it supports, and you can strongly claim that indeed it's trying to minimize its surface area, potentially for evaporation loss. And so this experiment provides us with some intuition about why aggregate. It's to reduce evaporation and for survival of the collective. This is further reinforced in other colloquial observations. So these are after Texas floods. These are some worms in sewers. And this is one of my favorites. Even in sulfur caves, the scientist goes down where there's nothing, not even oxygen. And the only thing they find over there are these worm blobs, suggesting that aggregation is a perhaps very robust strategy for survival from many kinds of environmental assaults. So here's a different kind of assault that we are going to challenge these worm blobs with. This is a plate, and we create a very robust temperature gradient from about 15 degrees to 35 degrees, which is within their physiological range of surviving. And we put a light and a camera. I'm going to watch what the worms do when you present them with a thermal gradient. And this is what we observe. So the top video is at lower light intensities. And when we place the worm blobs in the center, they migrate towards the cold. So they don't like the hotter temperatures. They're cold-blooded. They prefer to be in the cold area. But what's interesting over here at low light, they kind of spread out and individually move towards the cold and go there. But if you increase the light intensity just a little bit, and the light intensity is not affecting the temperature gradient at all, you notice that they move towards the left as a collective. So what we found is kind of a knob where we can now observe a really interesting behavior, which is collective movement in a blob, which is a really fascinating behavior because you have this aggregate of all these worms, and somehow they're making a decision and collectively moving. It's really unclear, or at that time was really unclear, how does a blob move together? how do they know to move, and how do they exchange forces to actually move to the left? Because you would expect they're all isotropic ensemble. They should just kind of spread out or not go anywhere if the information is uniform. So this is the question we're trying to understand. What is the rules of motility, of collective mobility in a blob, and how do they move? Here's an example that kind of gives us some clue. So here I'm plotting the center of mass of the blob as a function of time, and you see this is a big blob. You can see the center of mass moves. This is a big blob that can move, but this is a smaller blob, and that kind of starts to give us a clue. This is a more tangible system. And what we started to notice are these jumps. So you see that the displacement has these kinks. So it moves in a very jerky fashion. And I'm going to show you a video to kind of understand how it's moving and what is the behavior. Here's a quasi 2D geometry where the worm blobs are presented. And the reason we do this is you notice that the worm blobs are so dense, it's hard to see the worms individually and quantify them. We use kind of these quasi geometries and smaller worm blobs to track individual worms to understand their behavior. So here's a video of about 20 worms. And what we started to observe is that some worms at the front, which is the cold side, started acting as puller worms, and they're pulling the worm blob. And the worms at the back acted as, creating these transient legs and pushing the blob up, which we kind of call wigglers. So you have these pullers and wigglers. The pullers are generating the contractile force to pull the blob forward. And the wigglers kind of act to reduce the friction from the surface to enhance this transport of a 20 worm blob by 2 or 3 puller worms. So this is our hypothesis, and this is our observation. And we want to now test, is this possible? Is this indeed realistically the mechanism by which a worm blob may spontaneously break symmetry by some of the worms acting as differentiating themselves into 2 different gates, and you have this entangled ensemble to kind of provide traction forces. So we test this in 2 ways. The first is to ask, can a worm actually exert any pulling forces? To test that, we built this cantilever beam and provided pegs. And in this video, you'll see at 2 different temperatures, right? We want to see the forcing at cold water. So the worm finds a peg and starts doing some pull-ups. And it's actually much better at doing pull-ups than I am. You'll see it really, really deflect this beam. And once it's calibrated, we can estimate the pulling force because we know the mechanics of this beam. And in hot water at the same about 32 degrees Celsius, about 12 degrees different, we observe 2 things. One, the worm does not apply pulling force even if it makes contact with the pegs, which it does over 15, 20 trials of these different worms. But for the majority of the time, it also maintains a curved profile. So the colder worms naturally extend and kind of are in this relaxed structure so that they can extend forces. And at hot temperatures, the worms kind of coil up. And even if they extend a little bit, they won't exert forces. This is a summary of the forces, and we can see that in cold, the maximum force that these worms can apply are about 180, 200 micronewtons. If you do a basic calculation, that seems to be enough to pull a blob of 20 blob size if you estimate what the coefficient of friction is for these acrylic surfaces that they're moving on. So this is supporting our hypothesis that indeed it's possible that the worms can pull. The next way we tested, and this is where we teamed up with Dan, and some of you may be familiar with Dan's work in Smarticles, where they build these robots that are flappers. And effectively, this Smarticle can flap its 2 legs, but if you put it on a surface, it doesn't go by itself. And Dan and his team showed earlier this year in a paper in Science Robotics that if you confine them to force them to entangle, through these stochastic interactions, you find that the blob actually moves together. They refer to this as a cloud of entanglement because it's artificially entangling these Smarticles by putting this physical 3D printed ring around them. And they're moving towards the right because there's a light over here, and the robot, one of the robots, is kind of sensing and kind of holding itself rigid to create this symmetry breaking. So we wanted to use these to test our ideas. And what we did is we took one of these Smarticles, and for one of them, we programmed in at high light intensities a crawling gate. By crawling gates, these can actually break symmetry and move, and you can see that it's doing this in a alpha space. It's going symmetrically. And at low light conditions, if the light was less, then the smarticle would basically do a wiggle. And this is kind of replicating our behavior hypothesis in the worm blobs of pullers and wigglers. So these robots will act this way. The second part that we had to innovate is that these smarticles, you notice that in the previous experiments, you had to have a confining ring to create entanglement. Here, we wanted to really replicate some of the features of self-entanglement, and so we covered them with these hooks and these meshes so that they could entangle. And here you can see that they form a cohesive structure in themselves. And I'll show you a video towards the end to show that we can both, similar to the worm blobs, aggregate in the smarticle blobs, which we now call slobs, but they can also dissipate by changing their gait. So I'll show you an example of that control. So here's the aha moment. We have a slob collective. And if only the bottom worms wiggle, you see that the collective doesn't move. And if only the front robot pulls, you'll see again that the collective doesn't move, although it's an aggregate. However, if both are activated, so you reduce friction by the bottom wriggling bots and you have a symmetry breaking force applied by the crawling robot, you see that the entire blob moves. Now, obviously, these smarticle robots are not as sophisticated, soft-bodied, and complex as the worm. And here I have to add the caveat that really all models are wrong, but some are useful, and this kind of helps us test some of our hypotheses. Here's really a quantification of what I just showed you, that wiggle per crawl actually leads to finite displacement, but the other gates do not. Well, here's an example. If you let all the robots crawl, you actually see, similar to the behavior in the worms where they all move towards the light at low light conditions individually, you see disentanglement and you see the blob dissipating. So providing us with some design space. And here's a whole series of experiments where we can play with different controls and programming of robots. The point of this is now we have a test bed where we can test different collective behaviors to start to think, how do we program individual behaviors and organisms and how they can provide some useful functionality, locomotion, solid-like behavior, liquid-like behavior. And it's a really nice combination between organisms and robotic collective experiments. What we are excited about now, that was some of the next things that we are exploring, and really the worm blob is a fascinating gift that keeps on giving, is here we can put a worm blob in a maze, and there's food at the end. See that the worm blob starts in here. It sends out some scouts, and as soon as a few scouts reach the food, the big worm blob takes the shortest path. I'll play this video again. Notice that it hangs out in the bag. This video is over 8 to 10 hours. Scouts reach the food. As soon as scouts reach the food, within minutes, the worm blob always takes the shortest route. We have a whole suite of these experiments, but the mechanism is unclear right now, and we're evaluating it. Other aspects which are exciting is to think about three-dimensional behavior. So one of them is this ability to provide more mechanical functionalities. Here's a worm blob using different worms as arms to pull food, but here's also a worm blob in a 3D printed ladder to climb, really motivating some of our next explorations to think about these robots and worm blobs in unstructured environments and how they navigate things. Hopefully I've shown you a glimpse of how we think we've just opened a can of worms, and we have a lot of exciting things to explore. With that, I'll pause and thank you again for the opportunity to share some of our discoveries with you. And I'm happy to take questions.
Great job, Saad. Jeanette typed a question in the chat. Let me type her question.
I can. Oh, okay. Saad, really beautiful talk. Can you hear me?
Yes, we can hear you.
Thank you.
Okay. But so much, again, the density of your talk is the density of your worms. But I have a question about schooling behavior in krill. And I want to know, how can your understanding of information transmission in worms help in understanding how larger organisms with complex sensing and processing capabilities modify their swarm structures. The reason I ask is I want to know what information is needed to predict the nearest neighbor distance and orientation in order for krill to modify the shape of their school, which looks amoeboid, slow changing shape changings, which is in contrast to the near instant shape change form of fish schools that rely on visual signals, which move so much faster than hydrodynamic signals. Is that too complicated? I think that's why I wrote it.
Yeah, I think you're asking a very hard question then.
Yeah, I want to know if you can tell me what information I need to predict how a larger organism might orient or how close they need to be packed. Yeah,
so obviously I don't know the answer to that, but I can, so the single cells where the information exchange is, it's, I wouldn't say easy, but relatively easier in terms of understanding the signal generators and the sensors, because we can kind of simplify them to a mechanosensitive ion channel with a binary switch that triggers once a critical shear stress is detected. So it's a very simplified system, right? With the worm blobs, as you have multicellular system, which I recognize, I believe krill are multicellular, right? These are not single cells anymore.
Oh yeah, they're shrimp.
Right. So what happens is what we've also recognized with the worms and the collective decision making and adaptations is, as you saw with some of the temperature and light activities, you provide them with multiple information pathways. So in this case, the temperature gradient remained the same in both experiments, but we changed the light. And they collectively are computing and doing some control pathway and deciding their thresholds at which they will change their behavior. The point is that it's unclear how multiple modes of communication and information lead to a collective decision. And that's where it's a hard problem, because it's not as simple as just one fluidic wave. For most multicellular animals, they will perceive temperature, shear stress, forces, light, shadows, as well as chemical signals, which is the case for the worms, and I imagine for your krill, which I think makes this problem a little bit more complex to decouple how they're processing multiple modes and what are the thresholds for multiple inputs to make a collective decision. Does that help? So we are struggling with that, and I think that's a really cool question. And under certain confinement and certain regimes, we may be able to isolate all the other signals and just focus on one or two to understand that. But all times, everything is a very hard problem. So we'd have to kind of narrow things down a bit.
Yeah, but if I didn't, what I'm thinking is the rate of shape change tells you the type of signal that is being transmitted, meaning a school of fish changes almost instantly, so you assume it's a visual signal, while a school of krill changes slowly, more like your worms, which tells me it's a hydrodynamic signal that is a wave that's being propagated. Oh, okay. It would be hard to study in krill, just because they're in a big, giant ocean. Thank you.
Yeah, thank you.
Somebody else is asking.
Don't everybody shout at once.
Yeah, Saad, great talk. The first part of the talk, it seems like the density is really critical for getting them to communicate. How do they try to actively modulate their density? Like if I were those unicellular organisms, I would probably not want to spread past a certain density. What kind of things would make them go to this density where they're isolated? I mean, how much control do they have over the distance between neighbors?
Yeah, so if you see in this video, this is one example of their natural behavior, and both in the worm blobs and in the single cells, we've started to pick up patterns of we call it like an active pressure, but really they know when they're too dense, and they kind of find this critical density because they have local motion and think of them as kind of active particles. There is a certain density beyond which the other particles cannot come in, and I don't I don't have the final answer on this is not out, but I don't know how they're actively doing it, but there is a behavior under certain conditions in which you can start to predict the density. And the way we do this experiment is really map out the diameter of these aggregates that they form, both in the worm blobs and this, and at specific conditions of environments, we can find where they form these aggregates. To give you a more concrete example, so here I had put about 200 to 300 of these single cells, and you saw that majority of them, about 90%, are in this one cluster. But what would happen if I put in, say, 2,000 of these cells? You wouldn't just see one, you would actually see them form different packets, but you would start to see a periodicity in these aggregate diameters, again, revealing that beyond a critical density, they don't expand to too much. Does that make sense? So
yeah, they basically try not to go to a low density, you're saying. They make efforts to maintain.
Effectively, yeah. Yeah, give them, if you watch the natural cultures as well, you'll see that they're not infinitely forming one big giant hulk, but they will form patches. And you can change also the gap height. So if you put them in quasi 2D, and you'll start to see that there is a packing that they like because of their ellipsoidal bodies as well.
Is it they actually can sense other worms, or do they just want to have physical contact with objects? Like if you put like half dead worms and half live worms, would they still think that they're surrounded by their by their friends?
So when you talk about worms, you're not talking about these single cells.
Sorry, not worms. These just look like worms. I'm talking about the cells.
Yeah, I haven't done that experiment, but yeah, that's I don't know about the dead ones just because. But the sensing part is a little complex because they also physically touch, but at the contraction level and the density during their contractions, kind of there's a complexity between the fluid dynamics and their actual physical touching. It's a good question. I have to admit, I don't have a very good answer to that, but these are all just observations.
So maybe they may not actually touch. Oh, Lin Zhang has a question. I guess I'll read it. The communication between single-celled Spirostoma is fascinating. Do we know anything about the chemical process involved in the communication?
So our hypothesis is that the spreading of information is really too slow for any chemicals. And to the best of our knowledge, there is no chemicals that they're shedding that might act to spread information over centimeters that quickly. We suspect it's purely hydrodynamic because you have to ask what is the information being sent and how they're sensing it, and we've kind of answered both of those parts. So to the best of our knowledge, we don't suspect any chemical because you also have diffusion limits on these tiny molecules. Nothing can move from one cell to another over centimeters in milliseconds. Diffusion would be too slow, and even if it was actively convected, which we've done with particles, it just doesn't go that far.
David, I still have another question if nobody has one. But anyway, I'm still stuck on this.
People can type their questions in the chat.
Yeah, I'm still stuck on this krill schooling. So if I know that it's a specific signal type, fluid mechanical signal, and I know the location of the sensors, and I know their swimming speed, can you predict the nearest neighbor distance and the orientation that would maximize signal You can!
So yes, with a small caveat. Yes, if we kind of know their Reynolds number of information.
I do.
And if we have enough biological data about their shapes and how they arrange themselves in either quasi 2D geometries or 3D, we can.
Wow.
We can build the same models that we have, which is effectively just to kind of do a collective simulation of active particles that are generating fluidic waves in a fluidic medium and calculating effectively the Navier-Stokes and the shear stress at every point, and then ask what is the critical shear stress that would trigger a particle and its orientation, and what is the sensing limit on that active particle.
Okay, I'll find you on campus, or I'll talk to you. Thank you.
Thank you.
Okay, it's about noon, so I think people are gonna head to their next activity. But we'll have a virtual coffee with Saad for a few minutes if people want to ask some remaining questions. But thanks everyone for coming.
Thank you.